We'll pass the screen over to Tim Shields now of NCATS to present Pharos, the IDG data portal. All set, Tim?
Yep. Unfortunately for everybody, I'm not an avatar, so you gotta look at me while I'm talking. Um, so my name's Tim Sheils. I'm a developer at NCATS Informatics Core, and I did actually started early on. One of my first days at NCATS was at a meeting discussing whether or not we should start with the IDG program. So I was partially involved for a little bit for a while among other projects. So I was kind of always lurking in the background as a silent partner for Trung and Raj, and then took over and then handed it off to Keith, who's also on the call, kind of heckling me on Slack in the background. So we're going to talk about Pharos, which we called the open source target illumination platform, and it was designed to be the front end, the user interface for the TCRD, which in itself was the entire bolus of data that the IDG had aggregated and brought together to try to develop these target development levels. So the KMC, the Knowledge Management Center, provided, besides Drug Central, the Target Central Resource Database and Pharos. So the database itself aggregated 78 resources, has expression data, publication data. There's a whole list. I don't need to read all of them. And then with this data, use different measurements that were available to determine the development level, which Jeremy and Anna have already spoken about, based on dark targets have little known information to T-CLIN targets where there is an approved drug available. From there, NCATS Informatics Group designed a user interface because having all this data is nice, but people did want to be able to browse it and view it and kind of see what's inside this database and then potentially gain further access. So a little over 20,000 targets, a little under 20,000 diseases, whole bunch of ligands and compounds. From all of this data and the different sources, we created a large list of different filters that were available, added some user functionality to be able to, sign in and upload and save lists, share that information. And then as the program progressed, we performed user research, and we're always giving demos and seminars to try to figure out what other aspects people were interested in seeing and enhance that, in addition to working with data generating centers within the IDG that were also generating new information. So we tried to incorporate as much stuff as possible. Here's just a list of some of the different data sets that were in the TCRD and are currently available with the package. So with all of this information, we wanted to operate Pharos as almost a shopping site where you're going to have information you want to find. You're going to want to get a list of targets related to that information, and from that, see what is available to them, maybe compare development levels, see if there's any commonalities, and hopefully potentially find targets that aren't necessarily obviously associated with your target, but may share commonalities and could be a target of interest. So we have the ability to search over a multitude of facets and different properties, and I'm going to move through these pretty quick. Obviously you can browse. Browsing through the entire genome in Pharos is not too helpful, but there's filters along the side that allow users to kind of stack different categories and different filters and find hopefully targets that they're interested in. From the filters, we have different types of filters. You can search within the filters themselves. Each one has definitions. We made a pretty strong effort to include help and definition panels wherever possible. You can filter by numerical properties, so novelty or a PubMed score, how much published data there is about it. And so really the idea is to take these 20,000 genes and using similar properties, narrow it down to hopefully a workable set. Each drug or target details page is divided into sections with a general overview, if there's approved drugs or ligands or active ligands available, some of the different disease associations, disease novelty. This is the 10X that Jeremy had brought up. There's pathway information. We worked with Reactome to incorporate their data as well. There's wiki pathway information. We pulled in AlphaFold models and used some PDB models to display information as well, the structures. You can filter and search and examine gene ontology terms. And so, each page just has tons of information that's available for users. Protein-protein interaction, we have the sequence details. It's a pretty long list going through each page. So to help with that information, there's going to be this question mark button that will explain where definitions come from, what the source is, and how to get more information. So we talked about the target development level. So this is just kind of a quick example of how a target progresses from T-dark, what the requirements are, what the cutoffs are, how to view the supporting data, and then how to get all the way to the targets with approved drugs. And then, this is a panel that pops out. It'll show you the definition. If there's more information about how a score was computed, we'll have that as well. So again, trying to really highlight where this information, where these measurements are coming from. And I mentioned IDG generated resources. So these are all groups that were part of the IDG. And this data is unique, right now to the IDG. So it's information that you can find here and get reagents, assays, cell lines, mouse models, different data sets that were all generated as part of the IDG. In addition to your list of targets that may be associated with the disease or have a drug approved for them, you can also find targets that interact with them. And as you perform some of this list analysis and compare different lists of targets, we have dynamic sections that'll show up to show, what the interaction details may be, what the association may be, what the activity may be, and it's varies depending on what properties of the list that you're looking for. You can find targets that are similar based on different processes. So right now, this one's just the Go process, but there's a bunch of different categories that you can then try to find targets that have similar values. And then you can read the common values, you can read the data that gives us these results. All of these lists are downloadable, so you can save a lot of this target information as well. And that's, we also updated some of the publication and components. So we've got our word cloud, an improved publication view. You can download publications associated with the target or the gene riffs associated with them. And then it'll also show you how recently this information was downloaded. We added new visualizations for the 10X data. We just had in PeerJ the 10X version 3 publication come out. And so this allows you to kind of highlight where your target is or where a group of targets related to this disease novelty is. So it's a really helpful way to take something that's a scatter plot and see kind of, it gives you a little bit more information about what you're seeing within that plot and a little bit more details about what it is that you're looking at. But the whole purpose from this though is we have this massive database and we'll show people all this stuff and they'll say, well, that looks great, but where can I get the data? So people are and especially even at NCATS for informatics, people just want the data. So we have lots of different ways you're able to get the data out as well. If you just want some data about a particular target or a particular section of it, there's a download button. It brings up a download query that you can build. You can pick and choose what information, what properties you want to get. It'll show you what the SQL query that we're going to run is, and then a quick sample of what your information is going to be. So once that's done, you can run your download query and download the information. We do tell people though, if you have tons of information, the database itself is downloadable. So you can download and load it yourself. And then these SQL queries come in handy because then it gives you a starting point as well. In addition to just the raw database, we incorporated, we moved from a traditional REST API where you would get different levels of information as you progress. So to get programmatically uniprot a session for a target, if you have a uniprot ID, how can you get drug information? So first you would have to get the target data to get our ID for it. Then, and this is an old version, none of these links work anymore. But then you would have to get just the targets and then get information about each target. So it was multiple stages and it kind of started to take a while. So we switched over to GraphQL. It was created by Facebook and operates more on some of the principles of interconnected information, like a graph database. So instead of having to make all these specific jumps in one query, you can have it follow this loop. So you have you pass it an object with the query. Actually, this is a better way to look at it. With what the query is, so you can pass in parameters and you say, I want the uniprot ID, the ligand counts, the name and value, and then give me the drugs associated with that, filter out the ones that are approved drugs, and then give me the names and activities. So this would have been, 1, 2, 3 queries to get that information, whereas here you can get it all in one query and page different sections of the result. So rather than us, the developers, kind of determining what information should be shared, this allows users to kind of pick and choose whatever information they want and really help streamline the process. And again, we have a detailed documentation page and a sandbox. You can try these queries out at pharos.nih.gov/API. That's all I've got. As usual, there's a ton of people to thank who are involved within NCATS and with the IDG, and we've got the team itself. This was back in the fall 2023 where we had a NCATS informatics retreat. So Keith, who does the majority of the development work on Pharos now, Jessica Binder, who we snagged from UNM and is now helping us work on kind of a next stage of target information, and Evie Mate, our informatics head. So I'll pass it back to the group and can answer any questions as well.
Thank you, Tim and Keith Kelleher, for just the amazing work on Pharos over the years. It's really an impressive thing. I have one wrap-up slide, which I think we can fit in under the time limit if Mackenzie doesn't blow the whistle or something.
No, absolutely.
Okay, so basically, I think you've gotten the message, audience, for over a decade, the overall message, for over a decade, but with some work continuing, a diverse and talented consortium of experimental and computational researchers has focused their efforts on illuminating understudied human proteins as potential new targets for pharmaceutical therapeutics, using a wide variety of methods and approaches. The resources generated are available to continue facilitating and accelerating discovery, with efforts underway to sustain these resources to continue and maximize their benefits. For those of you wishing to learn more about IDG, these links will lead to an extensive set of documentation, publications, and the resources themselves. Please also feel free to contact me and I believe the others on this panel and other IDG contributors to learn more and perhaps explore making effective use of IDG data resources for your research. And to echo the others, thanks to all the IDG leaders, contributors, and advisors over the years whose enlightened and dedicated efforts produce these resources, pun intended. So thanks, everyone.
Thank you all so much. Before we leave, again, thank you so much for IDG for participating today. I just wanted to highlight our next webinar will feature an overview of the Human Reference Atlas version 2.1 from HubMap. So please tune in. And if you are unable to attend either part of this or you have colleagues that haven't been able to attend today, we'll have this recording up on YouTube along with the coordinating slides. So thank you again for everyone at IDG for participating. Thanks, everyone.
Yeah.